Hello, my name is Danya. I'm a third-year PhD student at the University of Oklahoma, and I will be presenting my poster, A Chemical Cartography Approach to Understand IFE Pathogenesis and Insights to Targeted Therapeutics. Influenza virus, or IFE, is a major contributor of respiratory illness, and each year 1 billion people are infected and there are 646,000 deaths. While IFE vaccination has been a major component for IFE prevention, it does not take into consideration the large diversity of the influenza virus as it has several different strains. Because of that, researchers have now become more interested in host-directed therapeutics. However, for that to occur, further understanding of IFE pathogenesis must occur. Hence, we present a chemical cartography approach that combines chemical techniques to develop 3D models of organs to determine where small molecules are located and their role in overall organ function and disease pathogenesis. In terms of our methods, we infected mouse models with the influenza virus, collected their lung tissue, and sectioned those into different pieces. We registered those into 3D space, extracted the metabolites, ran them on the LCMS, and mapped those metabolites in 3D. So we were able to develop these 3D models specifically showing viral load, so how much of the virus is actually present in both infected and uninfected mouse lung. As you can see, there's an overall difference in overall effect of infection, where specifically we were able to look at specific positions within that lung tissue and see that there was increases in viral load, specifically from blue to red, where it represents from low to high, that there is an increase in certain positions in infected mouse lung. In addition to that, we were able to look at specific chemical families, including isocarnitine and phosphocholine, where we see isocarnitine, this isocarnitine family member was found to be increased in specific positions of the lung tissue, and phosphocholine following the same trend to be found to be increased in specific positions as well. In addition, we looked at principal coordinates analysis plots so we can determine how different samples are from each other, specifically looking at how plasma relates to lung tissue. So circles that are closer to each other represent those that are similar based on their chemical profile versus those that are further apart or more different. Where plasma is in orange and lung tissue is in green, we see that there's two different clusters, which tells us that circulating chemical profile does not reflect local lung tissue profile. In addition, we looked at the effect of infection, where infected samples are in red and uninfected are in blue in both lung tissue and plasma. We see that there's two large different clusters, and we see that specific chemical families or samples are strongly affected by infection. This work is currently being done, where we will continue to explore positional differences in infected lung tissue, carry out further analysis of molecular networks, and carry out statistical analysis to determine statistical significance. Again, my name is Danya, and thank you for listening to my talk.